Hello, my name is Kyle Boehm, and thank you for coming to my poster. I will be talking to you about Drosophila melanogaster as a model organism for nervous system development. Firstly, Drosophila melanogaster, aka fruit flies, are a great model organism, which just means that research findings can be applied to other organisms, such as humans. Now, we study the evolutionarily conserved bone morphogenetic pathway and its role in nervous system development during growth. Now, the nervous system is just the communication system of the body. And it does this using chemical and electrical messengers, and the exchange of these messengers takes place at structures called synapses. Now, the synapse that we study is the fly neuromuscular junction, or the NMJ. This is because the fly NMJ is a glutamatergic synapse, which is structurally and functionally very, very similar to the synapses in the human central nervous system. Now, the method in which we examine the NMJ is we do something called an NMJ prep. So we'll take a third instar larva and we'll fillet it open and stain it with fluorescent antibodies. You can see in the bottom left-hand corner that the muscle will fluoresce green and the nervous tissue will fluoresce pink. Now, on the right image, you can see an individual NMJ. And when we zoom in, we see these little bulbous or button-like structures. We call these boutons, and this is what we quantify for our dependent variable as a way to measure growth and development of the nervous system. Now, the BMP pathway is partially responsible for the growth and development of these structures. And the BMP pathway is a retrograde signaling pathway, which just means at the synapse, the muscle sends a messenger across to the neuron. Now, we found previously in this lab that tau was a novel inhibitor of the BMP pathway, and it does this by phosphorylating something, which is a cellular on-off switch. Now, our hypothesis is if it inhibits the BMP pathway, if we were to remove it, we would see an overgrowth phenotype or phenotype with bouton counts over 20. Now, we did this using RNAi, which is RNAi interference, or RNA interference. This essentially degrades mRNA, which is the instruction manual for protein synthesis, and prevents the protein from being made. So we did this to tau, and we used tissue-specific drivers to do this in the muscle or the neuron, and we would expect to see an overgrowth phenotype of where it functions. So you can see in the graph that by removing tau in the muscle, and we see no change. But if we remove tau in the neuron, we see the overgrowth phenotype. So that indicates that it functions on the neuron side. Now, we know that tau has partners in other tissues like tricornered and GCK3. So we wanted to see if that was the case at the NMJ. So we did the same exact thing with RNA interference. And we saw essentially the same phenotype with loss of GCK3 and loss of tricornered. So that indicated that yes, there were partners. So we wanted to do something called epistasis to see the order in which they functioned, which essentially is pairing two things together and seeing which phenotype is expressed and which one is suppressed. So the two pairs that we did was we did a GCK3 overexpression and we paired it with loss of tau or loss of tricornered. Now in the graph below, you can see that the tau RNAi has the overgrowth phenotype, but when we pair it with GCK3 overexpression, we see that the overgrowth phenotype is suppressed. So that means that it functions prior to GCK3. Now, conversely, in the tricornered RNAi pair with GCK3 overexpression, the overgrowth phenotype is not suppressed, which just means that the GCK3 overexpression was suppressed. So the correct order in which they function is tau, GCK3, and tricornered. And that's essentially what I did this summer. Thank you for coming to my poster talk. I want to give a special thank you to everyone who helped make this possible, especially the Collar and Weaver SURF program. And if you want to know more about how tricornered actually affects the BMP pathway, then go check out my partner's poster, Patrick McGraw. Thank you for coming to my talk.